All right, I am going to move on to topic 7. Remember, we have 10 topics. Topic 7 is the cell cycle and mitosis. Topic 8 will be meiosis. And then 9 and 10 next week will cover principles of genetics. So we are almost done reviewing, guys. If you need any other type of organization, if you want me to send you any kind of list, like I did have a student ask, can you list out the chapters for each unit? I'm more than happy to do that. Just let me know. But I think, what I've given you in the handouts I've given you, it's a pretty good review to prepare for this particular AP test. So if you have all 10 topics, you've filled in all the notes, you've watched all 10 videos, I really, really think, and you've participated in the FRQs on Thursdays and Fridays, I think you are going to be golden when it comes to the AP exam, as long as you review back over those notes that you took. All right. So when we talk about mitosis and meiosis, you need to be able to differentiate between somatic cells and gametes. So remember, somatic cells are all body cells that aren't sperm or egg. So that's easy to remember. A somatic cell is going to be anything that's not a sperm or an egg. They have two sets of chromosomes, one from mom, one from dad. Somatic cells are the ones that are going to divide by mitosis. Gametes are your sperm and egg, and remember they only have one set of chromosomes, and the reason for that is because one egg and one sperm unite to form another living organism. They each only need one set of chromosomes so that that final organism then has two sets. If it was the other way around and a sperm or egg had multiple sets of chromosomes, you'd end up with a defective human, if we're talking about humans or any other kind of organism. Gametes are going to divide by meiosis. I'm going to discuss that tomorrow. So just remember, mitosis involves somatic cells, any cell that is not a sperm or egg. So skin, liver, hair, whatever. All right, it's important to know the phases of the cell cycle because in cell biology and in seventh grade life science, you probably just focused on this one little piece of the pie right here, which is mitosis, which is very important. But we need to look at all of these other things that are going on because G1, S, and G2, which are called interphase of the cell cycle, are going to set the cell up for mitosis. And either mitosis can happen too fast, it can happen too slow. A cell that is defective with DNA that is mutated or any set of problems can arise. It's interphase that is setting the cell up for whether or not it's going to divide. So if something goes wrong during interphase, it's kind of too late and things are going to get screwed up. That doesn't mean that there isn't a checkpoint during mitosis that has to be passed. But if you can remember, there are two checkpoints during interphase that have to be passed in order for a cell to divide. So we tend to forget about this because it's almost like, nothing's really happening. And within mitosis, you have your spindle and you have your chromosomes lining up and the separation of your chromosomes. It's lots of activity happening during mitosis. But this is so important, especially when you're looking at cancerous tumors and cells that are dividing out of control. So I just wanted to point that out. The interphase starts right here. See where my little mouse is? Right here. And then you have G1, which is growth, S, which is where DNA replicates. And I talked about that last week on Tuesday. And then you have G2, preparation for mitosis. And then finally in mitosis, you have cell division and then the division of the cytoplasm. And this is just kind of what I just went over. So I would know the order and just in general what happens in each one. In G1, the cell grows. S, DNA replicates, and G2, preparation for cell division. And then in the M phase, you finally have your mitosis and cytokinesis. You are not supposed to be asked any questions about identifying pictures. You won't be asked questions about identifying pictures. What I would like for you to have memorized, though, is this event right here, metaphase, and this event right here, anaphase. Metaphase, chromosomes are lining up in the middle. Anaphase, chromosomes are being pulled apart. Why are these two the most important phases. Well, the reason is because there is a checkpoint that makes sure the sister chromatids are each attached to their own spindle so that they can get pulled apart. Remember, if sister chromatids don't separate properly, you end up with a new cell that doesn't have enough chromosomes. And with mitosis, that's not a huge issue. That cell would just undergo apoptosis and die, whatever. But in meiosis, it is a big issue because that one cell could either be a sperm or an egg that unites with another gamete that becomes a living being, and that is set up to then have a defective number of chromosomes. So hopefully that made sense what I just said. Email me if it didn't. It is just very important that the kinetochores are going to attach to chromosomes properly right there. Then this just goes through each phase. Like I just said, metaphase and anaphase are the most important. Metaphase lining up in the middle, and anaphase the separation of sister chromatids. All right, so the entire cell cycle is going to involve checkpoints, and a checkpoint is going to control whether or not a cell is going to continue on with mitosis. So in number 1 on your handout, I gave you a long list of vocab words. I'm going to go over each one of those vocab words. You can just jot something down for each one, make your own list, or just kind of go back through this presentation. It's on Veracross, and just check those off. But I think drawing a simple picture to represent mitosis, so what you would want to do is go back to this picture right here. I wouldn't get too much into the details. I would focus on knowing metaphase and anaphase. So that's question number 1. Question number 2, how is the cell cycle regulated? Well, the cell cycle is regulated with checkpoints. And those are control points where stop-go signals are going to regulate the cell cycle. All right. Then on number 2, you're going to want to also add, so you're probably going to need a separate piece of paper for this because I don't think I gave you enough room. How is it regulated? Well, it's with checkpoints, but knowing the individual checkpoints is also important. So the G1 checkpoint is basically going to make sure, it's the most important one, it's going to commit a cell to being a dividing cell. It doesn't automatically pass it into mitosis, but it basically is a checkpoint where it's like, all right, you are destined for division. Let's see if you can get through these other 2 checkpoints, and we'll let you go ahead and divide. If a cell doesn't go through the G1 checkpoint, it goes into the G0 phase, where it's basically like a stop, where it's in a non-dividing state. So liver cells and, muscle cells, muscle cells, liver cells, and nerve cells usually are in a G0 state. Right, the G2 checkpoint, let's look at where G2 is. G2 checkpoint. Oh, conveniently, it is after G2. Let's see what's going on there. G2 checkpoint is going to make sure that DNA was replicated correctly, and it's going to make sure that there's no mutations. If the DNA replicated correctly, there's no mutations, and the cell size is appropriate, it's going to let the cell pass finally into mitosis. Then in the middle of mitosis, there's a metaphase checkpoint that makes sure that the chromosomes are attached to the spindle correctly with kinetochores. And this just talks about going into G0. So if a cell does not pass the G1 checkpoint, it's not destined to divide, it goes into G0. That's your nerve, muscle, and liver cells. Okay. This is going to talk a little bit more about the M spindle checkpoint and the kinetochore. Remember, a kinetochore, so if you can see my cursor right here, the kinetochore is kind of the teal colored thing right there. It's on that microtubule from the spindle. It attaches to that chromosome because the goal is for these 2 chromatids to separate and to each one to go in its own direction for the new cell. So that's what the M spindle checkpoint is really important for. So how do cells, or how does the cell pass each checkpoint? There's internal regulatory molecules called kinases and cyclins. And I like this picture a little bit better because it gives you a better visual. But anyway, kinases are proteins that control the cell cycle, and they are activated by cyclin. When kinases and cyclins join together, they form this thing called an MPF, and it's the MPF that then pushes it past this checkpoint right here. And this is mostly for the G2 checkpoint. There's other regulatory proteins, but this, for whatever reason, College Board thinks that these cyclins and kinases are the most important. And maybe it could be because some cancer treatments are targeting kinases and cyclins. It could be that. All right, but just so you know, how is it regulated? It's regulated by the checkpoints, and the checkpoints, the cell passes the checkpoints with regulatory molecules, and the G2 checkpoint in particular, passing the G2 checkpoint going into M, would require kinases and cyclins to come together to form what's called an MPF, maturation promoting factor. All right, cool. Okay, that's really all I have. It says, what happens if the cell cycle goes unchecked? That's number 3 on your document. If the cell cycle goes unchecked, the cell divides out of control and you could get a cancerous tumor. So the answer to that is cancer, basically. What are all the ways that cell division is regulated so that cancer does not occur? So you have your checkpoints. And then the other thing that isn't in this, this presentation is anchorage dependence. So the cell needs to be anchored down, not free floating. And then density dependence. If cells are too crowded, there's going to be a signal for them to stop dividing. So that's 3 things: checkpoints, anchorage dependence, density dependence. All right. And so those are the answers to those 4 questions. I don't think it will go too much more into that. I will talk about P53 and tumor suppressor genes. I'll talk about that a little bit tomorrow before I go into meiosis. But for now, what I'd like to do is, if you see the FRQ that's on your paper, if you can stop the video, time yourself for 20 minutes. This is kind of a hard one, I think. When I went through it, I thought it was kind of difficult. So if you can stop yourself 20 minutes, and then I'm going to go over the answer. All right, so it looks like at the beginning of mitosis, you have sister chromatids, and they're held together with proteins. 2 chromatids held together with proteins. An enzyme called separase, which is so clever. I wish I could just high-five the scientist that came up with that name. It's an enzyme that literally separates the 2 chromatids during mitosis. In cancer, separase is overexpressed. And that makes sense because if separase is cleaving 2 sister chromatids, that means that it's easier for the cell to divide, right? And cancer is an overdivision of cells. So what scientists are hypothesizing is that they can slow or stop cancer by blocking separase. And so how are they going to block separase? Well, they found this compound called sepin-1 that appears to inactivate separase in vitro in a test tube. All right, cool. So sepin-1 is supposed to inhibit the growth of cancer cells by inactivating separase. So what they did was they added increasing concentrations of sepin-1 to different types of cancer cell lines. So cancer cell lines probably include cancer cells from different types of tissues. And what you need to know is a treatment for one cancer cell line may not be a very great treatment for another cancer cell line. I think we talked about natural killer cells. And if this is totally going over your head, that's okay. I don't even remember if I really brought up natural killer cells in this class this year, since immune system isn't quite as covered. But natural killer cells are a type of white blood cell that kill cancer cells. And so scientists were like, great, we just inject cancer patients with natural killer cells, which was a great idea. But they found that it only works with blood cell cancer, and not other cancers. So you just need to know that just because something works for one type of cancer doesn't mean it works for another one. So that's why with this table you see A, B, C, D, E, F, or however many there are different cancer lines. You're going to need more of the sepin-1 to actually have an effect. So what they're trying to see is for each cancer line, what is the sensitivity to SEPin1? So how much SEPin1 does it actually require to kill 50% of those cancer cells? All right, so that's what we're seeing here. You have cell line EFGHJKLMN. So it looks like in figure 1, so this is what kind of confused me a little bit. It looks like in figure 1 we're dealing with cell line A, B, C, D. All right, and what they're trying to see is as you increase the amount of SEPin1, what happens to cell viability? Does it actually kill the cell? And what you should see is there's a trend as you increase SEPin1, cell viability goes down in all of the cultures. Then in the table, you see cell line EFGHJKLMN, and they're trying to see what is the relationship between the sensitivity, like what's the relationship between SEPin1 and separase? How if you need more SEPin1 to kill to keep the cell from dividing and to kill the cell, does that mean there was more separase there, or does that mean there was less separase there? What's the sensitivity? And I think this kind is a little bit confusing because you would think the more sepin required to kill the cell, the more separase was probably there, but that's actually not true. The more separase that is in the cell, the more sensitive that cell line was to the treatment. Which actually that does make sense because SEPin1 is acting on separase, so that actually kind of makes sense. But it looks like the more separase there is, the less amount of SEPin1 was required to cause a 50% cell death. Okay, so we're going to answer the questions. Identify the point in mitosis at which separase cleaves the protein complex that holds sister chromatids together. When during mitosis would separase cleave the protein complexes that hold sister chromatid pairs together? So what you would want to put there is anaphase is when sister chromatids completely separate, but it's at the end of metaphase where they're actually cleaved and pulled apart. And so you could have said metaphase or anaphase, but it really is actually the end of metaphase. You could have put either one though. So that's one point. Explain how the progression of cells past sequential cell cycle checkpoints and the activity of enzymes such as separase is controlled by interactions between two major groups of regulatory proteins. They're wanting you to talk about the cyclin-dependent kinases on that one. So what you would have need to say is basically what I said on that slide about cyclins and kinases and the MPFs that form. Cyclins and kinases come together to form an MPF, and that's going to induce the cell to move to different stages. Okay, I'm going to move on. Identify an independent variable, and then identify a control. You are guaranteed to get this question on your AP exam. You're guaranteed to get those error bars and to state whether or not something's statistically significant or not, and that's easy. If they overlap, it's not. If they don't overlap, going left to right, it is. If anyone's confused on that, please reach out to say something. And then identifying a control. A control is a basis of comparison. So if you're testing a treatment, the control would be no treatment, right? So the independent variable is either the cell line or the concentration of SEPin1. The control would be, I would say, if I was a rater, I would take no concentration of treatment, but they're saying a very low concentration of treatment. So no or low, I would take either one on that. So those are 2 points total for that one. Based on the data, describe the general relationship between the sensitivity of the cells to SEPin1 and the concentration of separase. All right, so what you need to say is the more separase there is, the more sensitive the cells were to SEPin1 and the lower the dose required to kill 50%. All right, then it says use the figure 1 data to calculate the ratio between the amount of SEPin1 required to kill 50% of the cells and that is most sensitive to the compound and the cell line that is least sensitive to the compound. So these are the answers. There is a 1:3 ratio between the amount of SEPin1 required to kill A, the most sensitive cell line, in which approximately 10 micromolar kills 50% of the cells, and D, the least sensitive cell line, in which approximately 30 micromoles kills 50% of the cells. So what I would definitely pay attention to in these questions, when I first looked at this question, I used table 1 to answer this question right here. So even I can get mixed up, and this makes me so angry that they don't separate these questions out. Why are they going to label it C, ask you a question about table 1, then ask you a question about figure 1 in the same question? It infuriates me. But I guess I guess they're just trying to see, well, can they read something and know exactly what's being asked? Can they look for detail, which is actually important in science? So maybe I can see their reasoning a little bit. All right. We have this one right here and this one right here. So this one's asking about table 1. This one's asking about figure 1. All right. So let me know if you have questions on that one. And there's the answer. All right. The scientists hope to test SEPN1 in clinical trials with human cancer patients. Remember, these aren't humans that they've been doing it on. They've been doing it in vitro, which means they've been doing it in test tubes. Sometimes things can be a lot different when you take the entire human body systems into consideration. The scientists claim that one way to test the efficacy of SEPN1 treatment will be to obtain periodic samples of the patient's cancer cells and determine the percent of cells in metaphase. Periodically taking samples and determining what percent are in metaphase. Why would they do this? How could this give them an idea of the efficacy? So remember, and this one, I take issue with this one because I think that the question should have said anaphase. I really do. But they don't. They say what percent of the cells are in metaphase. When I first read this question, I thought, oh, well, the more cells that are still in metaphase, the better, because that means that the separase hasn't effectively cleaved the chromatids and they won't move into anaphase. However, College Board is saying something different. If the compound is effective, the rate at which the cells are cycling should decrease because SEPN1 should decrease the amount of active separase in the cells. If the cells are cycling less frequently, then fewer cells should be in metaphase. So I kind of read into it a little bit differently, and I'm curious to know if any of you did too. If you're looking at this and you did say that as your answer, what I said, email me just so I know I'm not the only one that was thinking that way. I thought if they're still in metaphase and they're not actually separating yet, then it's been effective. But apparently the cell can sense if there's enough separase there to begin with and won't even go into metaphase to begin with. So I feel like they left that out. Maybe they didn't. If someone wants to correct me on this and tell me where I should have found that information, let me know. I know I'm the teacher, but still, there can't be other AP teachers that aren't like this too. Sometimes we get these FRQs and we're like, what are they thinking? But sometimes it's maybe just a word that I missed. All right, so that is it for today. Less than 25 minutes. Hopefully, you're getting what you need for the AP review. I am going to send out the instructions for taking the AP exam. I just feel like it's a little soon right now to do it, and I don't know if I have enough, if students have enough sense of urgency to actually do the assignment. So I am waiting until Monday, May 11th, and I'm going to have you do, instead of a Labster or HHMI, you didn't know this, but you did your last Labster or HHMI for me yesterday. So instead of having a Labster or HHMI, you're all going to do the demo for taking the AP exam and then reading through the instructions that I send you. So, all right, tomorrow I will cover meiosis and I'll do the exact same thing. I'll send a video, I will send a note-taking guide, and I'll have a practice FRQ at the end. Let me know if you have any questions. Talk to you later. Bye.